My background is I'm a stem cell biologist, but I'm also a neurobiologist who has a longstanding interest in neurodegenerative conditions, Alzheimer's disease, Parkinson's disease, stroke, spinal cord damage. I've been in the field about 25 years now, and my major interest around cells has always been the use of cells in cell-based therapies in humans. I'm not interested in curing mice or rats. All of our work has really been driven toward the clinic. So I spent 15 years at King's running a big stem cell group here in London. And then for the last 6 years or so, I was chief scientist for GE Healthcare. And now I'm outside of the business. I'm a freelance, enjoying life for a change instead of traveling and running major groups. And so now I'm a senior consultant to GE Healthcare, but I'm also a consultant to a number of other organizations, mostly within the regenerative medicine stem cell space. So let's get started. What David's asked me to do is kind of give you an overview of what are stem cells, what is cell-based therapy, and what's the future. So this is the organization that I currently consult for. It's called Life Sciences. It's a component of GE Healthcare. And it's a really big portfolio. You know, you start out over here, really, again, our big interest is in cell-based therapies. This doesn't seem to be working. Anyway, if you start out over here with research instruments, this is basically scientists working at the bench with the idea of taking what comes from the bench into the clinic. So we have this huge portfolio of research tools. GE itself has this massive interest in regenerative medicine and cell-based therapies, as I'll show you in a moment. Moving farther to your right, molecular diagnostics, or what's called precision diagnostics, is becoming a really big deal. And this is, rather than a histopathologist or a pathologist sitting in a microscope looking at slide specimens from, let's say, 10 chaps who are being diagnosed for prostate cancer, and he goes, yes, yes, no, yes, yes, no. All of those guys get the same drug. It's now precision diagnostics. That is now taking a section from a tumor and maybe looking at 50 or 100 different markers per section, and more intensively interrogating that particular form of cancer, and then tailor-making the therapy for that particular form of prostate cancer. So the idea is to stratify patients based on molecular fingerprints or molecular phenotypes of a particular form of cancer and tailor-making the therapy for that. And this is really going to revolutionize medicine, because it's estimated that something like only 20% of people actually respond to a given drug. And yet you're giving the same drug to everybody, which obviously has healthcare economic implications, and it drives up the cost of healthcare worldwide. And the therapies are getting more and more expensive, as I'm sure you've been reading in the newspapers. And so the idea of giving people drugs that are more likely to be effective in their particular form of the disease is really important, and it should help lower the cost of healthcare. And then clinical imaging. So GE is one of the world's leaders in what we call diagnostic imaging. So, magnetic resonance spectroscopy, CAT scans, ultrasound, SPECT and PET, et cetera. All of that becomes really important when you start thinking about putting cells into patients. Okay, so what are stem cells? In essence, stem cells are cells that have the ability to replicate more or less continuously throughout life. So if you start over here on the far left, when you're developing tissues inside an embryo, each of those tissue populations is replicating. There are stem cell populations within those, so that give rise to the liver, give rise to the heart, give rise to the brain. And then as development proceeds, those become more and more restricted, such that by the time the baby is born, you don't have stem cell populations in most of your tissues. And those tissues are basically fixed, okay? And you will have the same number of cells more or less for the rest of your life. But you still have stem cell populations that are really, really important. So how many red blood cells do you make every second? How many? Approximately 3 million a second for your whole life. Think about how many cells that is. What happens when you eat? The food goes through your body. It shears off the epithelial lining of your intestine. So every day you have to rebuild your intestine. What about your skin? Every day you rebuild your skin. For some of us, it's, we rebuild our hair. For some of us, not, unfortunately. It's estimated that if you live to be about 80, there are probably 8 of you standing next to each other of all the cells that you've ever made in your life. So these populations are incredibly important. They keep us alive. If your stem cell populations stop in your gut, you're in serious trouble. Okay? So what we've done is we've kind of exploited these cells and tried to figure out how to use them in disease states, in injury, and try to repair damaged tissues. Now you can kind of do this from within, and I'll show something at the very end about that, or you take the cells out, you try to expand them, and then you put them back, okay? So there's a whole bunch of different stem cell populations within here which are incredibly important. Where it says adult stem cells, mostly we're talking about hematopoietic stem cells that reside in your bone marrow that give rise to new blood cells every day. And when you do a bone marrow transplant, it's ostensibly a stem cell transplant. Because if the stem cells from the donor do not engraft into the host, that person will die, okay? Because you've irradiated their own bone marrow to get rid of their leukemia. And if the new cells don't go back in, you don't get reconstitution, you can't make blood, you will ultimately die. There's another population of cells within the bone marrow called mesenchymal stem cells that 20 years ago we were interested in using them to make bone and cartilage. These cells, as we've discovered, have amazing immunomodulatory properties. And so people are using them now to in autoimmune disorders like multiple sclerosis and lupus and others, because the cells appear to have capability. They secrete factors which seem to dampen down the immune system. So we have these cells now that are becoming more and more and more versatile. But there are adult tissues that don't have cells that we can do that with. So you do this with a liver. With a healthy liver, you can get some expansion of cells, but you can't get anything from the pancreas. So if you get type 1 diabetes and your beta cells get wiped out by your immune system, we can't take cells out from the pancreas, expand them, turn them into beta cells, and put them back. So for a number of tissues, we have only one real source population that we can turn to, and that's human embryonic stem cells, which are made from human embryos. Okay? And this is really controversial, not so much in the UK, but certainly in the US, this is extremely controversial. So this is in vitro fertilization. You start with a fertilized egg at the very top left. You basically allow the fertilized egg to develop as it would in utero. Actually, it's not even in utero, it's an ovario. And you, normally on about day 3, the embryologist will look down the microscope and pick out the best 2 embryos based on morphology. So these are high quality embryos at day 3. Those get implanted, and the remaining embryos are generally frozen, so you don't have to go back and superovulate the woman again. And you have embryos that if the first 2 don't implant, you can thaw them, put them back in, and try again. So many years ago, IVF was developed here in the UK by Bob Edwards, who won the Nobel Prize for this. And it was recognized very quickly that if you stimulated a woman and you got 10, 20, or 30 eggs, you had to fertilize all of those. You freeze those embryos. It's a shed load of embryos. At King's, where we did about 500 cycles a year, we estimated we had 35,000 embryos in liquid nitrogen. So the logistics of this are really ridiculous. And so the regulators said, 5-year cutoff. You can freeze embryos for 5 years, and then at the end of 5 years, you have to make a decision about what you're going to do with them. And generally, couples would have them destroyed. They rarely donate to another couple. They could be donated for research. And a really brilliant chap, first Martin Evans in 1981, showed that you could make from a mouse equivalent of an embryo at day 6, you could make these cells that you could just grow forever. And you could turn them into all sorts of interesting tissues. In 1998, Jamie Thompson in Wisconsin did this in humans for the very first time. And what this involves, basically, is taking what now is called a blastocyst, and you harvest the small population of cells. Is it showing up? Yeah, there we go. It's a small population of cells down here called the inner cell mass. This is what turns into you and I. And this is what you look like when you're 6 days old. Okay? It's about 50 cells. And what Jamie figured out, again based on Martin's seminal work in the mouse, was that if you remove those cells, you put them into culture in very sophisticated culture conditions, you can sometimes get something to grow that goes from being the original 50 cells to something much larger. Okay? And if you take this even further, what you develop is what's called an embryonic stem cell line. And what that is a line of cells from a single embryo, so it has a single genome. It's not a universal genome, it's the genome that embryo had. But you can expand these cells ad infinitum. So to give you an idea how expandable these cells are, this is called H7. This is one of the first cell lines that Jamie made back in 1997. It's been used by probably 1,000 labs worldwide. The number of cells that have come from this single embryo, which would have been destroyed anyway, would fill this building. I mean, these cells are really unbelievable. You know, they're like the ever-ready bunny. They just keep growing and growing and growing and growing. And the important thing is that with the right conditions, they are, at least in principle, capable of being turned into any cell type in the body. I mean, think of the power of that for regenerative medicine. I can grow 500 trillion of these cells and then turn them into something interesting, like this. So those are not cells that have been taken from a heart. Those are cells that in Cardiff we learned how to turn them into cardiac cells on an unlimited basis, really on an industrial basis. They beat just like your heart does. They have all the cell types that you would find in a human heart. They have ventricle cells, atrial cells, and cells that are nodal. They beat at about 70 beats a minute, and you can just grow unlimited numbers of these things. And then you can take the whole thing apart with enzymes down to single cells, freeze them, ship them all over the world, thaw them, plate them back out, and they turn back into what I just showed you. So they'll reform themselves back into a syncytia, and they'll start contracting again. This is work that we did in Cardiff and GE, and we did this for making heart cells for pharma companies to test their compounds on, because a lot of compounds get withdrawn because they have unforeseen cardiotoxicity. But you could use these cells for therapy, and people are. So let me back up for a second. So that's human embryonic stem cells, and GE is a human embryonic stem cell company. We've been very open about this since I joined 6 years ago. But, these cells are not genetically matched to the person that you're going to put them in. So unless you either put them into the brain or the eye, which is somewhat immunoprivileged, or you kind of figure out a way to hide them from the immune system, they're going to be rejected, or you're going to have to put people on immunosuppressive drugs. Okay? As a way of potentially getting around that, a very brilliant chap named Shinya Yamanaka, starting about 10 years ago now, said, okay, what keeps these cells, these embryonic stem cells, down here, what keeps these cells in this embryonic state, or what we call pluripotency? And so he and his team had a big look, and they discovered that there were about 20 or 30 genes that were only expressed in those cells that were not expressed in other cells, even progenitor cells, even hematopoietic stem cells. And so one by one, and then in pairs, they started putting each of these genes into skin cells, adult skin cells. And what Shinya was able to show that with 4 genes, he could take a skin cell from any of you guys in this room, put those 4 genes in it, and with a frequency of about 1 in 5,000, he could generate basically this from an adult cell. That is, he could take my little graph back here, and he could drive it this way. So now you can make the equivalent of human embryonic stem cells, but now they're genetically matched to you. There are something like 6,000 of these lines, and there are companies being set up who will make you your own cell line. And remember, you can grow these things forever, and you can turn them into any tissue you want. So if this young lady needs some heart cells, we could make heart cells. If you need new retinal pigmented epithelium, because you're getting macular degeneration, you could make that. You could make liver cells, you can make muscle, you can make skin, you can make bone. She also won the Nobel Prize, I think 3 years ago for this, because this is just revolutionized stem cell biology completely. And the idea that you can actually make the equivalent of embryo cells from a fully mature adult cell that was never going to be anything other than the fibroblast or a skin cell is really just revolutionized this field. So again, importantly, whether these cells are called induced pluripotent cells or IPS, importantly, whether it's from ES cells or from IPS cells, you can literally grow these cells. We've had cell lines that we made in my labs at King's in 2002, and we've been growing them and growing them and growing them and growing them ever since. So they expand ad infinitum, they retain this completely undifferentiated state, they make no commitments to any particular cell type, and then you can drive them toward whatever cell type you want. So right now in the clinic, there are people here in the here in the UK and in the US making retinal pigmented epithelium and treating patients with various forms of macular degeneration. There are at least 4 or 5 clinical trials at the moment, which have gone through phase 1 and are now going into phase 2. There's a trial of using these cells in acute spinal cord damage. There's a trial in California where people have figured out how to make pancreatic islets. And in patients with really severe type 1 diabetes, these cells are being implanted in a kind of capsule, so they're hidden from the immune system, okay? And more trials are coming on board in Parkinson's disease and others, okay? If you look at cells so that's embryonic. Let's move on. So this is, you can make anything, so. So the idea is, okay, so the idea is you look at a particular clinical disorder. Let's say it's it's Alzheimer's disease or it's stroke or it's Duchenne's muscular dystrophy, and you say, what do I need? What kind of cells do I need, and where can I get them? Okay, and so most of us in this field tend to be generally pretty pragmatic about this, And for example, in type 1 diabetes, if you can't get cells from the pancreas, I can't turn bone marrow-derived cells into pancreatic cells because they can't do that, then you have to go to either ES or IPS cells. On the other hand, if you want to make bone, I don't need ES cells necessarily. I can make bone from cells that are within the bone marrow. So for many of us, and certainly for GE, the whole idea is that you figure out how you're going to take any cell that has particular therapeutic potential, how do you expand it to the numbers that are necessary, and then put it back in to the patient in a very safe way. Now, if you look at, this is a really small number of different clinical indications. When you begin to think about treating hundreds of thousands, millions of people all over the world who have these disorders, and all of us will ultimately probably have one or more of these, the numbers of cells that we're going to have to generate becomes unbelievable, absolutely unbelievable. And the technologies are just not there yet for this huge scale. You're talking about 100,000 liter bioreactors. This is a really massive undertaking for this field to really generate the potential that we think it has. There's something like 4,000 different clinical trials ongoing in the US, in Europe, China, Japan, and a few other places to address this. And soon you'll see that there'll be licensed products, that is, approved cell therapies from the FDA, from the EMEA, and others. So this is fundamentally going to change medicine because, for example, in heart failure, if you have a massive MI and you lose 30–40% of your left ventricle, it's never going to come back. And all the drugs in the world are never going to rebuild your heart. You're going to always have that giant scar in your heart, and you're going to be impaired for the rest of your life. If, on the other hand, I go back in and I put some of those heart cells that I showed you into the heart at the time of damage, I may prevent you completely from going into heart failure, or more importantly, take you from heart failure back to a better quality of life and better heart function. So we're not going to be treating the symptoms of a disease, we're actually going to be getting at the underlying pathophysiology of the disease, and in some cases, reversing it. So this is really going to totally change the way we do medicine. One thing that's driven this field in the last 5 or 6 years is something totally unexpected, and it's become just the wild, wild west. And that's called cancer immunotherapy. And there are a couple of different ways of doing this. The one I'm going to talk about is basically cellular immunotherapy. This is not using drugs. And basically what we're doing is we're taking the immune system and we're teaching it how to attack a tumor. Okay? Now, tumors in principle are very, very good at evading immune response. And that's why in many cases, people get sick and die from cancer because the immune system hasn't seen this as being an aberrant cell, non-cell. It has very good mechanisms for just suppressing the immune system. A guy named Steve Rosenberg, who was a clinician at National Cancer Institute in Washington years ago, said, we should be able to figure out how to train the immune system to go back and recognize this. Let's try to figure this out. And for the last 25 years, he's struggled in vain. Nobody believed him. Everybody had given up on this. And then he showed that it works. And basically what you do, there's a number of different kind of flavors of this, but in essence what you do is you harvest bone marrow or mobilized peripheral blood. You then separate out the white blood cells from that. And then you may genetically engineer them, or they may already be competent to recognize the tumor antigen. But in most cases, we have to engineer them to express a receptor for something that's on a cancer cell. We expand them, and then we infuse them back into the patient 14 days later. It's really simple. It's not growing, hundreds of trillions and billions of cells. It's about 50 billion cells per patient. And this works, okay? This is absolutely amazing. And I don't use that word very often. So in the first study that was published by Carl June and Bruce Levine at Penn, who we worked with, they took 5 patients who were in-stage B-cell lymphoma. They had weeks, in case of Emma, days to live. They'd failed all conventional therapy, weren't eligible for bone marrow transplantation, took their cells out, engineered them, grew them up in our bioreactor, put them back, and within 3 days, all 5 of them were completely cured. Completely. No evidence at all of B cells whatsoever. There is no chemotherapy, radiation, any other therapy in the world that gives you complete remission from cancer. It will always come back. To date, they're all still disease-free. Michel Sandelin, who is a good friend, and we also worked with him, his data was even more impressive. 12 out of 14 patients, complete remission using the same technique. This has led to I like David talking about investment, this has led to this just massive gold rush. And this is all phase 1 data. This is safety data. This is not even efficacy data. Pharma never ever goes in and buys anything from anybody unless they're in phase 3. And this was 5 patients. Novartis has licensed Carl's technology to the tune of several $100 million. They're building a massive facility in New Jersey to ship cells, from hospitals all over the U.S., take them to New Jersey, do all the expansion and modification in New Jersey, and then ship them back. Michel's work led to the formation of a company called Juno, and in less than a year, between an IPO, some deals, and some cash deals, they raised over $1 billion, and their market cap at the moment is on the order of $6 billion on the basis of phase 1 data. Every major pharma company in the US and in Europe is basically building a franchise around cancer immunotherapy. I've never seen anything like this in 30 years of science. I've never seen anything just like take over. I've been at probably about 6 or 7 conferences in the last roughly 3 months all over the world, and this is the dominant scientific theme now in the cell therapy space. Okay, so this is happening. The challenge for us, this has all been done in what we call liquid tumors. These are all blood cancers. The challenge now, we need to apply this to solid tumors, prostate, breast, ovarian, brain gliomas, et cetera. And that's going to be a lot more challenging because, again, I said cancer cells have a very good mechanism for evading the immune system, So what happens when these new white blood cells get there? Are they just going to be suppressed as well? So the data on that isn't in yet, but that's the next big challenge. But we think we can figure out how to do it. Okay, that's kind of the state of the art at the moment. Now I want to show you some stuff that's like the next generation of healthcare related technologies. So the last year I was at GE, I ran this program called Blue Skies. And what Blue Skies was is, you know, if 10 years ago somebody in GE decided that GE should become a cell therapy company, what should we do in the next 10 to 20 years? What technology should we chase for us to make the next generation healthcare products? So I had this team of about 50 people worldwide, some really, really smart folks, engineers, software, electronics, a bit of everything. And this is just a rough approximation of some of the technologies that we thought would be really, really useful. So microbiomic diagnostics is one of my favorite ones. As scientists, we're so arrogant sometimes. So for years and years and years, nobody paid any attention to the bugs that live on us and inside us. You know, the bugs that live in your ears are very different than the bugs that live on your face. They're very different than the ones that live in your mouth. Very different than the ones that live in your esophagus. Okay, so we're roughly 100 trillion cells. We're 10 times more bacteria. Okay, so we're 90% bacteria. And yet we never thought about this. And then all of a sudden people started publishing stuff on the stuff that lives in your gut determines your propensity to obesity, to type 2 diabetes, to inflammatory bowel disease, et cetera, et cetera. You can take people who have a really nasty bacterial infection called C. diff, Clostridium. And this does, for patients who have a really bad infection, you can give them all the antibiotics in the world and this stuff never goes away. And they basically live on the toilet. And they have an incredibly bad quality of life. They're very anemic. They're generally pretty malnourished because their gut can't absorb anything. And they're just in constant pain. If you take bacteria from, let's say, their brother or sister who's healthy, and you transplant it into the person who has C. diff, about 98% of patients resolve completely in the absence of bacteria, in the absence of antibiotics. It's called a fecal transplant, or a shit transplant, however you want to put it. You can take genetically engineered mice that are bred to be fat, and take genetically engineered mice that are bred to be lean. Fat mice never get lean mice never get fat. If you switch their bacteria, they revert to the other phenotype, completely overriding genetics. I mean, we never thought about this. And this, this is, I mean, now there's, microbiomic diagnostics. And that's just the gut stuff. We haven't even gotten around to the stuff that lives in your lungs, et cetera, et cetera. I mean, this is just amazing. And then the other big one for me, and I'll talk a little bit about this, I'm running out of time, is machine-brain interfaces, okay? Putting electronics into your brain. But the one thing that we decided we would start on first, and this is a project that's still ongoing and I'm still involved in, is the use of what's called additive manufacturing, and part of that, and David talked about this, is 3D printing, okay? So can you print tissues and organs? So right now we're doing really simple stuff. We're not even printing yet, so we're making ears. So these are basically molded collagen, and then you spray paint some cells on them, put them in a reactor for a while, and then sew them on, and they work. Tracheas. My colleague here in London, Mark Lodell, he takes, for people, like a woman who had extremely bad tuberculosis, and her trachea on one side of her body was basically totally necrotic, and they were going to take her lung out. And the surgeon said, well, she's young, and this is really going to screw up her life. So what Mark did is he basically took a trachea from a cadaver, and he stripped the cells off of it using enzymes and detergents, so there were no cells left from the donor. Took some of her bone marrow cells, put on the trachea, put it in the reactor for a few weeks, implanted it, she's perfectly healthy. Okay. So I'll skip that. So the reason we got into this is because if you look at organ transplantation, it's clear that most people would never get transplanted. If you look at this graph, the number of people who need transplants is 10 to 1 that ever get them. And that includes living donor kidney transplants, okay? And if you look at the demographics, it's pretty grim, you know? So it's tissue type or organ types along the top, and then the time to engraftment. The number of people that are awaiting transplants. You can see basically if you don't get transplanted within the first 6 or 9 months of your disease, you're never going to get transplanted. The numbers go up and then they go down because people die awaiting transplantation. You know, but in many cases, like if we go back to the heart, in many cases you don't actually need a heart transplant. If you're in heart failure, what you need is a new piece of left ventricle, right? You don't, the whole heart, at least immediately, you know, a couple of months after you've had a big coronary, you know, the heart's still in pretty good shape. So all you need is that piece of muscle put back that's been damaged by your heart attack. So our idea was you basically take normal tissue. This happens to be a lymph node, but it could be a piece of heart. And you basically look at the cellular organization under the microscope. And not only do you say smooth muscle, you know, endothelial cell, fibroblast, et cetera, but what you do is you convert this into basically a computer-assisted design file called a CAD. You feed the CAD into the 3D printer, and it prints this. Okay? I'm serious. So we mapped this out, and the only bit that we're missing from a GE perspective is the fabrication step, the printer. So we're working with the world's largest printer company to try to see how reasonable this is. Can we really do this? And we think we can. It's technically challenging, but because if you're going to make a piece of tissue like this, and about 1 centimeter thick, it's about 800 million cells, and you're going to have to print that in 2 hours without damaging the cells. But watch this space, because this is coming really fast. What about brain repair? So right now, you know, if you have a big stroke, there's not a hell of a lot that we can do. If you're developing Alzheimer's disease, we don't really have any tools. Parkinson's disease, we do have some medications that work. We have deep brain stimulation, and we have transplantation potentially, because we already know that we've done that with human fetal tissue. But maybe one of the things you don't know, you actually have stem cells in your brain. And right now, probably by the time you leave today's lectures, your brain will have produced some new neurons, they will have migrated and integrated back into your brain, and you will have a memory of this event. For those of you who've been awake and you know, and don't have memory loss, okay? And so, you know, this is work we did back when I was in California almost 30 years ago. And so we were like, well, what happens in disease? So this happens to be a woman who had had a really massive big stroke. And this is about 6 days after she'd had her stroke. And so we thought, well, let's have a look and see. And what we saw was that here's the region I showed you where the stem cells normally sit, and they make, you know, let's say maybe they make 100 new stem cells a day. What we saw was this train of cells going up into the region of damage. Okay? And when you look at section by section, she had millions of these things. Millions of these cells migrating into a region of damage. What does it mean? I don't know. You can't track these. We have no imaging capability to track them. And even if she had survived, I have no idea what happens when those cells get there. What do they do? We know some people recover from stroke. They do get some function back. Is that because of this? So together with a few pharma companies, we're trying to see if A, we can understand this better, and B, can we develop small molecule drugs that basically stimulate this process without having a stroke? So could you give a drug like this to somebody early in Parkinson's or early in Alzheimer's disease? Rebuilding the brain from within. Okay. 2 more slides very quickly. So this is Eddie Chang, who's a neurosurgeon at UCSF. And what Eddie's doing is recording from the brain of somebody with epilepsy, but at the same time he's recording function. So he's trying to map out the brain delineate the seizure area from other high real estate areas like speech recognition, the motor strip, et cetera. And he's using 1,000 electrodes. And so for 2 weeks, 24 hours a day, he's recording from 1,000 individual electrodes and acquiring all this data. Can you imagine how much data that is? And then you got to process it, right? So he's got like 40 people in his group, and 20 of them are, bioinformatics and engineering types. And what Eddie has done, he's mapped out how our brains respond to sound. So if you look over here, there's a group of electrodes that are really, really strongly positive for da, ka, pa, ra, really hard consonant sounds. You see that here, but they don't respond to these sounds, but a whole another group of cells over here do respond to oo, ah, e, ah. Okay? So he's figuring out how our brain processes language. And the experiment he's doing, and I haven't heard the results of yet, is what if I stimulate this area at the same amplitude and frequency that the brain's responding to sound with? Do you hear sound in the absence of sound? Okay. Turn this around, imagine this is a speech area, and this is you saying ah, ka, da, ra, pa, but now you're a stroke patient, and this area of your brain has been destroyed. Can I take this information and move it over here? And now with the stimulator, if I stimulate at the same amplitude and frequency, can you then regain speech? Because speech is not here, speech is up here. This is cool, isn't it? So Eddie's consortia got 71 million of the first 100 million that Obama put up for this brain initiative. And his team, just his team alone at UCSF, got 26 million. The guy's brilliant. Last but not least, here's the surgery of the future. So you've got a tumor, it's inoperable, T cells don't work, so you need something better. So rather than slicing you open and going in and rummaging around, we send the nanorobots. So these are made of DNA. For those of you who are metrically inclined, it's about 50 nanometers in diameter. So they're nanobots. And basically, it's in a closed configuration. And it's carrying toxins or drugs or enzymes or whatever you need for your cancer. And it's got 2 sensors. And so both those sensors have to engage with the target. And when they get there, it opens up and it delivers the payload. So this may sound trivial, but my friend Idor Bachelet, who did this work in Israel, has already shown that he can eradicate tumors in his favored experimental animal, which is a giant cockroach about that big. So he injects them with tumor cells, and then he injects the robots, and the robots go in and they kill. So in 20 years, 50 years, you won't need physician surgeons anymore. You'll just need some robots. Okay? So with that, David, I'll stop, and thank you very much.
Any questions? Okay, Gareth.
Gareth Wong, thank you very much for, wow, this is tour de force. I just wondered the costs of investment in terms of, is there a big difference from stem cell to all these other gene therapy and, and then also time frame, compared to medicine, et cetera? Thank you.
Yeah. Right now these are all still very experimental, and the cost of production are high. I don't know. There's the first gene therapy ever approved in Europe. It's called Glybera. It was approved a few months ago, and it's 1.4 million euros per patient. Per patient. So we think roughly, I don't know, maybe Julia knows, but I think roughly the cost of making T cells for immunotherapy is several $100,000. But with automation, with further investment, I think automation is going to be the really big key. And then we're still very experimental. We don't know how many cells we need. We almost always kill the patients in the process of doing this because the cells are so potent when they go in the patients have this life-threatening condition called cytokine storm. The cells secrete so much interferon and tumor necrosis factor that the patients all have to go back on life support. But then they come off 3 days later, and when it works, they're cured for life. So, right now, these are all very experimental, and they're going to be very expensive, and they're going to be mostly for people in developed countries, obviously, but we have to get around that. We have to make these things available to everybody in the world at a cost that people can afford. But that's going to take time and automation. Sorry.
Hi, Stephen. That was really interesting. I just wondered what your thoughts were on the regulatory pathway, because obviously there's an issue around quality control, around small trials. It's very personalised, so how you get the repeatability. So I just wondered what your thoughts were on how you see these products going through and being approved, whether there has to be some sort of change in the landscape.
Yeah, I mean, we've been heavily in discussions with the FDA, as have everybody else. I mean, I was at a conference here yesterday in London, and there was a whole session on regulatory approval. The FDA has been very open to expedited approvals, particularly for the T cell stuff, because they just see it so transformative compared to drug-based cancer therapies. But when I've gone and talked to them about what's your opinion on 3D printing, they're like, Stephen, we haven't even figured out how we're going to regulate cells, and now you're already on to the next thing. So that they will come out with some guidelines probably within the next 5 years around 3D printing. So it's it's an evolving space. Most of us who are in this field are actively engaged with the regulators and trying to help them as they help us. So it's a definitely two-way interaction, but they've been very open to these things.
Hi, Stephen. Thanks for...
Oh, okay.
I'm Terry. Tour de force, brilliant. I always love hearing you. You never disappoint. However, moving on. I'm thinking about the decay of cells, where is it telomeres shorten, and that's how we age. And it strikes me, being a person who's not as young as I used to be, that research in this area could be helpful to stop us oxidizing or whatever it is.
Yeah. Well, my friend Aubrey de Grey, that's for those of you who don't know Aubrey, he's a very interesting Cambridge chap who believes that we could live to be 300 or whatever. And one of the approaches that he looks at is other drugs that can rebuild your telomeres, for example. I'm not saying everything that we do we have to transplant something new back in. A lot of what we want to do, particularly in the brain, is use conventional drugs to try to stimulate inherent endogenous stem cell populations. And so I think there's a whole bunch of different ways you can approach this. I mean, I could talk about this for days, and there are just a number of strategies that one could use to try to regenerate tissues, rejuvenate tissues, with cytokines, with small molecules, a whole bunch of things. We need to go to break, don't we?
Yeah, maybe there's, I think, a couple of other quick questions. There's one at the back, but we do need to break soon. Yeah.
Okay.
Hi. So this is a very self-interested question.
Okay.
What about 3D printing organs that have structures, but you have a genetic disorder which gives you a screwed up structure of your organ? So, for example, I've got my GP's mentioned to me more than once heart transplant, which has been rather worrying. But over time, my heart degrades in shape. Mm-hmm. So does that mean you could? So obviously, you could, I suppose, get somebody else's stem cells to create a 3D printed heart out of that. But in theory, would you be able to use your own heart?
Well,
cells?
So there's a couple of ways around this. I mean, heart's a little bit difficult because it's vascularized, and so I could make all the relevant cell types from embryonic stem cells. So I could make the cardiac cells, I could make the endothelium and the smooth muscle cells that you need for vessels. I could make the fibroblasts, but your body will see those and reject it. What I could do is I could take skin cells from you, turn them into iPS cells, and make the equivalent cells. And we've actually just transplanted the first patient in Japan using her own cells. It costs somewhere on the order of $2.5 million to do that. So it's not ready for prime time yet. But in principle, you could. And that's really the idea. I mean, how many of you have seen Fifth Element? Remember Leilu? Remember she gets printed? Well, actually, she gets knitted. But that's the idea that, it's like having an old car, a part wears out and you put a new part in. Or in the case of Leilu, if you need something really significant, you build it. And I mean, there are already people doing this. I mean, there are people printing like little liveroids And so this is not science fiction. This is I think, easily within the next 20 years, you'll be able to print replacement tissues. So hang in there. Hang in there. Well, it might be sooner. It depends on investment. Yes.
Hello. Hi, Stephen. So my question is to do with sort of like the capitalist elements in where this research goes. So you mentioned like the T cells are in the hundreds of thousands and some of these other therapies are in the millions. So will it not be a case of the business mindset overtakes the imagination that can be applied to these technologies? So whilst you could come up with great things, these companies want a return of investment. So is it going to be a case where the future is kind of like the rich old people's diseases that will be catered to, and like the poor young people who perhaps can't afford these therapies, their diseases will be skipped over?
That's a very good question, and I think equality in healthcare is a really, really big deal. Surprisingly enough, a lot of the companies now that are in this space are going after what are called rare orphan disorders, and most of these are kids, surprisingly enough. And so, like right now, nobody's going after really big, cardiovascular disease, Alzheimer's disease are the really big ones. And there's very little work in either of those with the exception of some transplant work in the heart. A lot of these companies are going after orphan indications where the numbers of patients are small, but it gets them skin in the game, it gets them moving along, it gets them investment. So I think it'll be a mixture of all sorts of things. But my big fear is that it's just so expensive that it only gets done in Europe, it only gets done in the US, and it doesn't get done in the rest of the world. And I think you have to convince the pharma companies and the VCs and others that has to happen. And we're working pretty hard on this, particularly even a company like GE.
Mark Friedman, organic chemist. Is this working? Yes.
Yeah.
Mark Friedman, organic chemist. The neural stem cell work. You mentioned that there is so far no imaging capability for the tracking of what's going on there. What do we know about that? What's the problem there?
Problem is we don't have the imaging capability to be able to see the cells because we can't, because they're born in the brain and they stay in the brain. We can't label them, which is the conventional way people track cells, is they load them up with gadolinium or iron nanoparticles and then use MR, or you put a PET tracer in them or something like that. We can't physically put anything into the cells because they're stuck in the brain. So what I've argued for is we actually make PET tracers that recognize a cell surface protein on neural stem cells in the brain, and then we could track them. Problem with that is it takes about $300 million to develop a PET tracer. And the return on investment is not very good. And I haven't been able to convince my bosses that this is something that we should do. So
there's scope for more innovation ahead.
Yeah, there's the universe. It's infinite innovation is required.
So we're going to break. We're going to come back in 15 minutes. So that's not very long. But if you want to stretch your legs, grab some of the refreshments outside, chat to some of your colleagues. Let's start again at 3:45. So thanks very much to Stephen. That was really fantastic.
Thank you. Nice to meet you. Cheers. Thanks. Yeah, it's fun.